So let's explore the role that pH has in biological organisms. And in order to do that, we have to look at the effect on pH on the molecules that are in the cell. Now this figure is showing a protein, and this protein is folded in a particular way, and that folding determines its function in the cell. Now, proteins can be denatured, and you can see that denaturation results in the unfolding of this protein, and it has also lost its function that has been associated with its shape. So pH, a particular pH, for example, can alter the shape and function of this protein. What I want to do is explore on the next slide of how this is so, and also explore the importance of buffers. Now, buffers minimize pH change. And so in the context of a cell, that would mean that this protein, for example, that is folded in its appropriate way for its function, that a buffer would minimize the pH change to prevent this happening, which would be the denaturation of that protein. So buffers minimize changes in pH. Buffers are weak acids that don't completely deprotonate in water. So let's go over some of the terminology. Hydrogen ions are protons, right? If you have a hydrogen atom, that is a proton and an electron. If it is a hydrogen ion, it's just a proton. So hydrogen ions are protons, and therefore pH is measuring the hydrogen ion concentration, but you can also think of it as measuring the proton concentration. Now, if a molecule takes on a hydrogen ion or a proton, it's said to be protonated. And if a molecule loses its proton, it is said to be deprotonated. Okay, let's go back to this idea of buffers not completely deprotonating in water. We're seeing two forms of carbonic acid here, which is a buffer. This is showing it in its protonated form, and on the right of this equation, it's showing it in its deprotonated form. Notice that it has lost a hydrogen ion, it has lost a proton. Now let's take a look at this buffer in this solution. You can see that some of the buffer is in its deprotonated form, meaning it is capable of binding a proton, and some of the molecules of this buffer are intact and they're still in its protonated form, and therefore can lose a hydrogen ion if it needs to. So let's say you challenge this solution with hydrogen ions, meaning you're adding an acid. Well, this deprotonated form can bind those protons, and that would minimize the pH change. Let's say that you added a base to this solution. This intact form can donate a proton, release it into the solution, again, minimizing pH change. So buffers minimize pH change by their ability to bind protons, protonate, and release protons, deprotonate in a solution. So let's review the importance of pH and buffers in a biological context. Changes in pH are going to can change the folding of proteins. How they do this is that proteins contain groups on them called R groups. And these are groupings of functional groups on these R groups. And it is the interaction of these R groups within a protein that contribute to the folding of the protein. That is how a protein can coil and twist to take on its three-dimensional shape. So if you change the protonation states on this protein, on the R groups of these proteins, you can cause them to no longer associate with each other, and that would cause unfolding, as you can see on the right, the denatured protein. Let's look at the specific example here of two R groups. This one is showing that it is an acidic R group, okay, and it is negatively charged. And this one is a basic R group, and it is positively charged. I want you to think of what would happen is if the pH changed here and kind of forced this R group to take on a proton. It would no longer be able to associate with the other R group. It would disattach and it would unfold. So again, buffers minimize the pH change by minimizing the change in free hydrogen ion concentration. That's two ways of saying the same thing. And that can prevent in a cell, for example, proteins from denaturing if you get changes in pH.